This video is the part 2 of respiration, and it's to do with the third part of aerobic respiration, which is the Krebs cycle. So far I've talked about glycolysis, the link reaction, and that is in part 1 of the respiration videos that you can find on Dr. Biology. And this video is going to concentrate fully on just the Krebs cycle. So the Krebs cycle is the third stage of aerobic respiration, and it takes place in the mitochondrial matrix, and here reduced coenzymes and ATP are produced, and there's a series of oxidation and reduction reactions. Now, I'm going to only discuss what is in the AQA A-level specification. There are certain kind of parts of the Krebs cycle that are not gone into detail in the A-level. Now, if you want to look at it, then that's brilliant, but just remember, for your exam, they particularly want you to specify certain aspects of the Krebs cycle. So if you cast your mind back to glycolysis, you'll remember that from one glucose molecule you get two pyruvate molecules that will then go into the link reaction. And the link reaction will happen twice, and so the Krebs cycle, it goes around, as it is a cycle not reaction, goes around twice per glucose molecule. Okay, so if you remember in the link reaction, the acetyl-CoA, so coenzyme A, is a 2-carbon molecule. So for the first stage, what happens is that you can see the acetyl-CoA there, so 2-carbon, and that combines with oxaloacetate, which is 4-carbon, and it forms citrate, or citrate, which is 6-carbon. Notice that the coenzyme is released and it goes back to the link reaction to be used again. So to summarize the first stage, acetyl coenzyme A comes from the link reaction, which is 2-carbon, and it combines with the 4-carbon oxaloacetate and forms citrate, which is 6-carbon. So once we've made the citrate, it is then decarboxylated and dehydrogenated. Now, just a reminder, decarboxylation is where carbon dioxide is removed, and dehydrogenation is when hydrogen is removed. And the coenzyme NAD is going to produce reduced NAD, as you can see. So as we move through, decarboxylation occurs, dehydrogenation, the NAD takes reduced NAD away to produce a 5-carbon compound. Luckily, you don't need to know the name of that, you just need to know it's a 5-carbon compound. Again, here's a quick summary. Okay, so citrate and the carbon decarboxylation comes off, producing CO2 as a waste product, and the dehydrogenation occurs, and the NAD collects the hydrogen and to produce reduced NAD, so you get a 5-carbon compound. Now the next bit is crucial. Okay, so far we've looked at acetyl-CoA and the production of citrate when it combines with oxaloacetate. CO2 is released, reduced NAD is produced, and you've got the 5-carbon compound. Now we're going to look at the next stage of how a 4-carbon compound is produced. So the 5-carbon molecule is now converted to a 4-carbon molecule via the following processes. Okay, so again you get decarboxylation going on, okay, and also you get reduced NAD. In fact, 2 molecules of reduced NAD being produced, 1 molecule of FAD, which is another coenzyme, and most importantly the production of ATP. Now, the ATP is produced by the direct transfer of a phosphate group from an intermediate compound that's not shown, and I don't need to show it you, okay, to the ADP. So this is called, and it's very important you remember this, is when it's converted, it's substrate level phosphorylation. Unfortunately, there's a spelling mistake there, so I'm going to spell it correctly. So substrate level phosphorylation is basically just the addition of a phosphate group to ADP. So now the citrate, as you can see, has now been converted back into oxaloacetate, and it's back into the Krebs cycle. So it's allowing the constant cycling. Now, there's quite a lot going on there, and I've got a really I think a really good way of remembering all the NADs, FADs, ATPs, etc. But before I show you that, here's just a quick summary. Okay, so you can see from the 5-carbon compound, decarboxylation, so CO2 is released, and then the dehydrogenation happens twice. There's obviously intermediate things going on here, because then you're getting substrate phosphorylation going on at with the production of ATP, and then this other coenzyme FAD. But the most important thing is it's regenerating oxaloacetate. Okay, so this next bit is not my own work. I found it somewhere and it's kind of stuck. And it's just a way to remember where the coenzymes are and the ATP. And you can see the Krebs cycle. Okay, so you can see that it goes dinar, dinar, a, fa, na. Okay, so dinar is basically decarboxylation and the dehydrogenation. So you get reduced NAD and CO2 twice. Okay, you get ATP, the A, which is ATP, fat, which is the FAD, so dehydrogenation going on, so FADH is produced, and another set of reduced NA. Okay, so to summarize all of that, the products of Krebs cycle. So from 2 Krebs cycles, you get 2 coenzyme A's, which is reused in the next link reaction, 2 oxaloacetates, which are regenerated for use in the next Krebs cycle. You get 4 carbon dioxides, so dinar, dinar, twice, released as a waste product. You get 2 ATP, because remember it cycles twice, which is used for energy, 6 reduced NAD, which is going off to oxidative phosphorylation, and 2 reduced FAD, which is going to the same place. So in a nutshell, that is the Krebs cycle. If you go on and do higher levels of biology, you'll find that this is quite simplistic. There are many kind of intermediate points in the Krebs cycle, but for your A level, this is what you need to know. Please do look at my next video, okay, which will be part 3, which is oxidative phosphorylation, better known as the electron transport chain. I hope you've found it useful, and I will speak to you soon.